So thank you so much for joining today. I don't think that Dr. Zoghbi needs any introduction to the MECP2 Duplication Syndrome community. As you all know, she identified mutations in MECP2 as the cause of Rett syndrome back in 1999. She's been an integral player in the duplication syndrome research community. She discovered that bringing levels of MECP2 down reverses many of the symptoms found in the animal model. And she's pursuing a therapeutic approach using antisense oligonucleotides. And she's going to give us an update today. This webinar is being recorded. Unfortunately, we can't see or hear the participants, but we welcome your questions. Please put them in the Q&A and we'll address the questions at the end of Dr. Zoghbi's presentation. Thank you so much for joining us and for making time. I know you're crazy busy, so this means a lot to the community.
Thank you, Monica, for inviting me. Thank you. Thank you for all that you do for Rett Syndrome Research and MECP2 Duplication Research, and thanks to all the community who is with us today. So I'm gonna start by sharing my slides, and I hope everyone can see them.
Yes.
Sounds good. So maybe, Monica, you may want to mute yourself because there's an echo. Okay, great. So today I'm going to share with you all the steps we're taking to move our preclinical work to the clinic. And I realize there may be some people here who may be new to some of the research. So I will take maybe 5 minutes just to introduce some past information that everybody probably knows, but just to put us all on the same page, then move to all the steps that we have done or are doing or we need to do to really get us to the clinic. So I think everybody knows the features of the MECP2 duplication syndrome. My entry into this disorder was really from a mouse model we generated in 2004 that had progressive neurological problems, including premature death by 1 year of age. And Ann Collins has made this mouse, and this made us suspect that duplication will cause this disorder. And then a year later, Linda Van Ash described boys with the MECP2 duplication syndrome. And as you know, there is a lot of genetic work here at Baylor. This is one example that shows that these duplications are really different sizes. They begin in different regions. Some of them have triplications. So obviously the levels of these proteins are gonna vary. And the only two genes duplicated in the region are IRAK1 and MECP2. Now, you heard probably from Davud last week or a couple of weeks ago how we're trying to really refine the studies to predict the level of the protein in each of these duplications. That's work he's doing. We also know that triplications are worse off than duplications. And we also know that decreasing the protein, of course, causes Rett syndrome. And if you have 50% of the normal level, you have a disease slightly milder than Rett. We've done this in animal models, but still gives neurological syndrome. So this protein, one has to get it as close to right as possible. So we started with the idea, can we reverse Rett syndrome in this mouse model? And just again, to bring everybody to the same page, we collaborated with Ionis. As you know, the DNA makes RNA, which makes the MECP2 protein. The antisense oligos are small pieces of DNA that bind the RNA. And when there is this mix of DNA, RNA, there's an enzyme that will degrade the RNA, and that will lead to less production of protein. And back in 2015, Hezi Steinberg tested ASOs in adult mice and showed that it reversed all their neurological features. Here's an example of the duplication mice. You see them less active. They don't stand on their hind legs as much. They don't enter the center as much. You can see the difference between a healthy mouse and the MECP2 duplication mouse. But when we give them the ASO, they behave very similarly to healthy mice. So this was exciting, and I'll finally just summarize one important piece of data. we wanted to see if we started the treatment really, really late, 8 to 9 months of age, when at that date in these mice, they're having daily seizures all the time. We wanted to see if we did that, do we normalize the EEG and do we stop the seizures? And the answer was yes. If you start the treatment, at least in mice at 9 months of age, 8 or 9 months of age, you see here this abnormal EEG normalizing and all the seizures stop. So that really gave me hope that this is something, if we start in people with duplication, is going to be helpful. Of course, the goal is to take what we've learned from the animal models and move it into clinical trials. And I know every one of you was waiting for that. But MECP2 is really unusual because of the dosage sensitivity. And it's the first time that a trial will be initiated on a protein that's dosage sensitive. So we had to take multiple steps to really prepare for the clinical trial. And today I'm going to tell you about these steps and how much we have accomplished. So the first thing one needs to do is make a humanized mouse to provide the proof of concept that you can titrate the dose and monitor the dose. The other thing we had to show that if you happen to overcorrect, if you lower MECP2 too much, is that reversible? And the next thing we need to track MECP2 levels. And so you need a biomarker that track the levels. And we're working on that, and I'll share that with you. And last but not least, we have some new data that shows us how much correction do we need to do to see some benefit. So today, this is the outline of the talk and all the material I will be sharing with you. So first, let's talk about the humanized mouse and the proof of concept data. Our original mouse had one mouse gene and an added human gene. So when we used the ASO against the human gene, we only eliminated the human gene and the mouse gene protected the mouse, so we could never overcorrect. But in the humans with duplications or triplications, the two genes are really identical. The two copies of the human genes are identical. So if you use an ASO, you're going to decrease both of them. And this is why it's important to create a mouse model that's very similar so that you can ask, can I titrate the dose of the drug so that we can stay within a safe range? So for that, we created a new mouse model that had the two human genes, but no mouse gene. This way you are sure that anything you see is from really mimicking the situation in a human. And these are the mice we generated, and we showed that they have almost twice, a little bit, maybe more than twice of the MECP2 protein level. And this is work again with Yao Shao and Hezi Steinberg. And just to show you here the data, you'll see that the animals have very similar features to the original duplication mice. You know, in Rett, the brain is small. In duplication, it's slightly increased. So the brain weight you see here is slightly increased in these mice. These are mice that have a little bit more anxiety. So you'll see them here. They don't like to be in the open arms. The open space makes them anxious. They spend more time in the dark space compared to healthy controls, and they don't enter as much in the open arm. So you see the signs of anxiety. We know if we put them on a rotating rod, they spend more time on the rod. It's almost like a repetitive perseverative effect. So they have all the features we saw from the original mice. So once we validated the humanized mice, that really they're a good model, we then wanted to use a new injection protocol that is more similar to what will be used in the clinic. Our original study, we used a pump where there was continuous infusion of the ASO. That's the paper published in 2015. But now for the proof of concept work, we need to put the injection, one-time injection, and we need to put it in the spinal fluid space in the ventricle and see, does it get to all parts of the brain? How good is the distribution? And luckily, there's an antibody to the ASO, and you can see here that when you don't use the antibody, you don't see any signal. It's blank. There is actually a brain here, but you'll see in the brain that received the ASO, you see a signal throughout the brain telling you that it is really getting to all parts of the brain. And now we ask, okay, now that it got to all parts of the brain, do we lower the RNA? Do we lower the protein? So we put the level here at 1. Of course, these are duplication mice, so they have twice normal. And you see we can normalize it in all brain regions. The RNA decreases in the cerebellum a little bit less than other brain region, but it still decreases. Then when we monitor the protein, you see reduction in the protein in all brain regions. We don't see it in the time frame we looked here in the cerebellum, but because the RNA is decreased, we know if you were to follow it for a longer period, there will be a reduction. Now we ask, now that we have a good model with high level of the RNA and protein, can we use different doses to titrate the dose? And I want you to think of MECP2 as similar to blood sugar. If somebody is a diabetic and you give them too much insulin, the blood glucose will go too low and the patient will go into coma. So you titrate the dose of insulin for a patient. And this is the same way. We want to titrate the dose of ASO to see the ideal dose. And you'll see if you use too low, you may not get it back to normal, but the 250 and the 500 both reduce the protein enough to get it to within the normal acceptable range. So then we asked, if we now give the dose of the ASO, how long does it take for the RNA to get normal? When does the protein change? When does behavior change? And here we start with the duplication at 100%. When we give the dose of the ASO within 1 week, you see that the RNA went down, and you see it's still down, and then gradually it goes back up. On the other hand, if we look at the protein, you notice that the MECP2 protein very slightly decreases by 1 week, but it actually decreases by 2 and 3 weeks. So you can see the protein levels of MECP2 normalizing, but they're normalizing slower, or later than the RNA. So then we asked, how about behavior? Do we rescue behavior, and when does that happen? And remember, I told you these animals stay longer at the rotarod, but here you'll see with both doses, the sort of medium dose and the regular dose and the slightly lower dose, the rotarod reverses, and the animals are similar to healthy control animals when on this rotarod. Then we do a test of learning and memory. These animals have defects in their learning and memory. These are the controls. These animals tend to freeze more, tell us their plasticity is altered. On the lower dose, there's tiny bit of reduction, but it takes this dose, the 500 microgram ASO, to get it back to normal. So then they reach control level. So we tested all these behaviors and all normalized. The only behavior that actually did not normalize in these animals is the anxiety. And you'll see here, these animals enter less into these open arms. So an anxious mice will not go into an open arm. And you'll see that both the low dose and the regular, the therapeutic dose, neither of them normalize the anxiety. Now, there are two reasons this may not have happened. It may take longer for the anxiety to disappear, or because these animals were born with anxiety, because also with the genetic rescue, we didn't see normalization. It may take additional behavioral intervention to really help with the anxiety. We will see. So I want to summarize then that first set of data for the humanized mouse and the proof of concept. We gave the injection as the same way it's going to be given in a human patient, in adult, mature, fully symptomatic animals, and within 1 week, the RNA level normalized. The protein followed. In 2 weeks, it normalized. I didn't show you the data, but many of the genes that we know are altered in this duplication mouse, we think those are downstream effects of MECP2, also normalized within 2 to 3 weeks after and stayed normal for a long time. However, it took a while for us to see the behavioral benefit. The behavioral benefit began to appear at about 8 to 9 weeks. So there is a lag time between the time we give the treatment, we make MECP2 back to normal, and then we see a clinical, if you will, improvement. And this is really, really important. It is important for two reasons. One, it tells us when we design clinical trials, we're going to have to be patient and we're going to have to accept it's going to take some time for the plasticity of the brain to be corrected after we correct MECP2 level. It doesn't happen immediately. The second thing, it tells us that in the case, we treat and we overdose, we have ways at the beginning, we have so many targets that we're going to begin to analyze and biomarkers to tell us if we corrected just to the right range or we overcorrected. And that would help us with a new guiding dose, or if it's necessary to give an antisense to the antisense, it can be done to cancel its effect. So we'll come back and talk about this later. So now we provided the proof of concept that actually you can do this when the two copies are identical, and you can titrate the dose of the ASO, and you have a really nice timing of the sequence of events. However, we still wanted to know, just in case, each patient might respond slightly differently, just in case there's one patient that the level of MECP2 goes way down. If you go way down, you're going to get Rett-like features. We wanted to know if we overcorrected just because of the nature of the patient's response. Is it reversible? And for that, we now designed a new experiment. In this experiment, we use mice with only one copy of MECP2. That's their normal copy, not the duplication mice. Remember, in duplication, we're going from twice to one time. So we want to decrease it by 50%. But here we only have one copy. And we gave the same time of injection that eventually will be mimicking the injections done in humans in the CSF. And we collected brain tissue. This is again in adults, animals, 1 week, 2 weeks, and so on after, and measured the protein levels and the RNA levels. And here you look at the protein levels. So remember, these are animals who had one gene copy. This is not what will happen in real life. This is an experiment we're doing to see, in the worst case scenario, if you brought down the protein to almost zero, as you see here, what will happen. So you see that within 1 week, there was a slight reduction of the protein, but by 2 weeks, it's about 25% of normal. That was intentional. We gave a very high dose of ASO to deplete it, to really see what is the very, very worst case if we totally eliminated the protein, to where we knew here you have like 10% of the protein, and now we followed these animals. And this is what we saw. First, the animals stayed healthy for about 9 weeks. Again, I told you there's a lag time between the level of the protein reduction and the phenotypes. Now, we're looking at Rett-like features. Remember, the MECP2 duplications stay longer on the rotor rod, but Rett mice stay less on the rotor rod. They're the opposite. And you'll see here, we gave 2 doses, and you'll see both of them reduce the performance at 9 weeks. So it took almost 2 months and a week to see these features after depletion of MECP2. And then we kept following these animals to see, is it reversible? And the answer was yes. And this is, I should mention, work by Yin-Yang Chao. It's unpublished. And you see here that it took about another 13 weeks after we depleted the protein and the animals are symptomatic, but the nice thing, it reversed. You see here, they're back to normal. Then we asked, how about their activity? And the low dose did not really decrease, or medium dose, but the high dose decreased their activities. And you'll see here a reduction in the activity. And even that, I should mention, this is nowhere as severe as the Rett mice. But still, there is a reduction in activity. And 13 weeks, you still see it. Remember, the animals only had 1 dose. 21 weeks, you still see it at the high dose of ASO. And by about 25 weeks, it reversed. So features, just like the rescue happens at different times, features are reversed at different times. So I'm going to summarize the data here for you. We're not going to go through every behavioral panel. So here we took healthy mouse with a single copy of MECP2. At this stage, we depleted them from MECP2, and the protein was pretty much down to 10%, about 6 weeks of age. And we started seeing anxiety, motor and coordination, hypoactivity. We didn't see other features, so the features were limited. And I should mention, all the features were still milder than what typically is seen in Rett male mice, but they reversed. Here, the anxiety reversed. You can say, well, why did this anxiety reverse and the MECP2 duplication did not reverse? Two possibilities. As I mentioned to you, the MECP2 duplication, we may have needed longer. We don't know. The second possibility is that here we induce the anxiety in adult animals. In the MECP2 duplication mice, they had the anxiety because the gene was overexpressed from birth. So it could be something about early exposure to anxiety may take longer to reverse or may require behavioral therapy on top of normalizing the protein. But that was the first thing to reverse here, the motor coordination reversed, and then the hypoactivity reversed. So this told us that even in the very, very, very worst scenario, if we over treat and cause total depletion of the protein, it's still pretty reversible, and it's not as bad as the Rett male mice, and you saw the timing of the reversal. But we don't want to get to that point. What we want to do is really have a biomarker that as soon as we make a dose of the ASO within 1 to 2 weeks, sort of get an idea. Did we hit it right? Did we didn't give enough? Did we give too much? So how do you do that? The best way to do that is to get something that tracks with MECP2 level, that is sort of a barometer of where's the MECP2 level. And this is what we are working on. Now, biomarker development, it could be anything. It could be a clinical biomarker. And I think you've heard probably from Dr. Suter and Pelivan through the Ionis funded study that they're working diligently to find such biomarkers. Dr. Mariana Malatic-Savatic is looking for metabolomic biomarkers. These are small metabolites that run in the blood and spinal fluid that she's trying to identify them, which ones are responsive to MECP2 level, and her work is funded by the MECP2 Foundation and now more recently by NICHD through an IDDRC signature project fund. We are not doing either of those things. What we are doing, we're looking for what we call a molecular biomarker that is either an RNA or a protein, something that changes the expression of a gene because of the changes in the level of MECP2. So we want those that reflect MECP2 levels. And just to make this easy for you, I'm going to just give you some examples of those. So let's take first MECP2 level itself, because we want something that track with MECP2 levels, right? So here's in the duplication, you'll see that MECP2 levels are really high. And if we now give an ASO, if you give a control ASO, nothing happens to a healthy mouse, nothing will happen. But here's the duplication, has high level of MECP2, and you give the MECP2 ASO as shown here. At the control, nothing happened, but you give the ASO in green that lowers MECP2 levels, bring it back to normal levels, and you'll see that it tracks. At first it comes down, as I showed you before, and then it gradually comes back up. These are weeks. So within a couple of weeks, it's going to come down. So it tracks really nicely and then goes back up. So the question, can we have a marker, a biomarker, a molecular marker? Here are some examples. So here's an example of this gene where in the duplication, it's high, but when you give the ASO to MECP2, you see it now coming down, down, down. When the effect of the ASO wears off, it goes back up. So that's an example of a gene that really nicely tracks with MECP2 level. And it's normally high when MECP2 levels are high. But sometimes we have genes that are low when MECP2 levels are low. So you can see here a gene that's normally low when there's too much MECP2. But when you give the ASO, it goes back to normal. This is where normal is. So you can see it going back and then coming back down. So here's, you've seen two examples of what I'm talking about, a biomarker that tracks the levels of MECP2. And then here's an example of one that doesn't, because I want to show you what it looks like. This biomarker, this gene, is high in the duplication, but when you give the ASO, it sort of changes all over the place. You cannot trust it. You really want something that you look at the curve and it really mimics MECP2. So Samir Bejikar in our lab is really working hard to find as many of those biomarkers that are highly reliable and study and showing that they also track with the disease in patients, hopefully, and eventually those would be the ones that we would use, I hope, in clinical trials. The last part I want to share with you is new insight we gained to learn how much correction is needed to see clinical benefit. Do we have to go from 200% to 100%? Ideally, I hope so, because that's the ideal correction. But what happens if we get to 50%? And what happens if we get to 70% rather than 100? We overshoot. So I want to share with you data that we learned from a recent studies that gives us insight about that. And these data were designed to find what regulate the RNA. They're sequences in our genome that dictate when an RNA is made, how much of an RNA is made. And this one, we were interested in the adult brain by finding those sequences that sort of open up the DNA to allow regulation. And this is how we found them, which collaboration with Josh Wyeth's lab. We identified several regions that drive the expression of MECP2, that sort of control it. These are in the DNA, these regions, and how that DNA is open, allowing other factors to come and bind to allow them to control the level of the RNA. This is how we control the level of MECP2 in the brain. This big fat peak here is a promoter region, which means you have to have that to express MECP2. This was discovered by Uta Franca many years ago. What we identified are these additional peaks here. 1, 2, 3, 5, and 6. So we decided to check which of those actually control the level of MECP2. And to do that, we used CRISPR to delete each one of those one by one and learn which one of them are important for MECP2 levels. And you'll see here, we created these different mice, and you'll see that this is MECP2 levels here. You'll notice that when we deleted peak 2, MECP2 levels were reduced by about 30 to 40%, as you can see here in the brain. Whereas when we deleted peak 6, MECP2 levels were increased, you see that. And then we asked, well, what's important? Does this hold true in patients or humans? And for that, we used iPSC-derived neurons. And here again, you see this is healthy human neurons. If you delete peak 2, you'll see there is 50% reduction of the MECP2 protein. And if you delete peak 6, there is almost a 50% increase in the MECP2 protein. Now, this is interesting because I'm sure you're immediately connecting, wow, that's a nice way to decrease MECP2 levels, and that is true. And we're studying this peak. I don't have time to talk about that today. We're studying this peak intensely to see what regulated, to see can we use this knowledge to find yet another way to lower MECP2, but that's going to be work into the next few months to years. And this one here, now we have the evidence that these two are important. So given that we had mice that decrease it by 50 or 30% and mice that increase it by 50%, we asked, what do these mice look like? And here is, you see the data. You know, Rett mice have all of these features. Also mice that have 50% reduction of the protein have most of these features. But what we noticed that the mice that only lack 30% of the protein in their brains, so that's the peak 2 deletion, they don't have all the features. They only have they have hyperactivity, anxiety, and social deficits. So they have just a subset of the features. And on the other side, the mice that have 50% increase, they don't have all the features of MDS that you see on this slide. They have a subset of the features, the anxiety, hypoactivity, and learning and memory deficits. So what this tells us, if you start with 200% and you go to 150%, at minimum, you're going to rescue some of the motor deficits, some of the social deficits, some of the epilepsy. On the other hand, of course, if we get to 100%, we're going to get to normal. But if we overcorrect by 30%, you will get a little bit of hyperactivity, anxiety, and social deficit. So it is important that we carefully monitor so that we're within this range. And this is why our goal to really stay within this range, but it's also encouraging for both Rett syndrome and MECP2 duplication that even if we didn't reach 100% correction, we're going to get a milder disease if we reach even 50% correction. But again, we hope we get better than that. And the same with Rett. If we got partial correction, we're going to help some symptoms, I believe. So I think this is also encouraging. So now I want to summarize all that I've shared with you so that we can have some time for discussion. I hope that I convinced you that the ASO has good distribution in the brain using a strategy that will be mimicking the clinical administration of the drug. It can reduce MECP2 expression in a dose-dependent manner, just as you would use medicine for high blood pressure or diabetes, where you titrate the dose. And that the treatment rescues the majority of the behavior, except for the anxiety-like phenotype, at least within the duration of the study. We haven't done these repetitively, we just gave one shot. And then in case the ASO therapy leads to excessive lowering of MECP2, the effect is reversible. There are multiple candidate genes that are sensitive to MECP2 levels, and these provide an opportunity to develop biomarkers to track MECP2 levels for the clinical trials. And finally, even a small reduction in MECP2 levels is likely to make a difference and improve some features of the MECP2 duplication syndrome. So with that, I'd like to thank all the people who helped us get to this point. First and foremost, both the Rett syndrome family and the MECP2 duplication families. Obviously, the work on the duplication has been totally, really inspired by seeing the children affecting with this disorder. And I still recall Brody, my first MECP2 patient. And while unfortunately Brody is not with us today, I hope and dream of the day we bring treatment, both for all the children who deserve it now and to honor Brody's legacy and all the other boys that have suffered from this disorder and sadly passed away. I also would like to thank the Rett syndrome families and the MECP2 duplication families who participated in the clinical readiness trials. We obviously need both Rett males and MECP2 duplication to really know our boundaries in those studies. I want to thank you for your support for this research. I think many of you remember the 401 project that really was started to help us on April 1 to get the funds to fund Hezi, who started in our lab to begin to design the genetic rescue and reversibility that then morphed into ASO trials. That could not have happened without the 401 project, which is now, I think, folded through Cure MDS. At the time, the funding came through the Rett Syndrome Research Trust, and we're thankful for all of your support and your continued support for us to explore other areas. We're pushing forward with the ASO, but as you've heard me say about the regulatory element, we continue to push new strategies. We want to really do as much as possible for both of these disorders, Rett and MECP2 duplication. And I'm very grateful for Ionis for their support of preparing the ASOs for us and working with us to do all the right steps for the preparation for clinical studies. And more recently, they are now helping us with the biomarker development of the work that Samir is working on. And of course, NINDS and the IVDRC course here at our institute and our institute course are all contributing this work. It takes a lot. So a lot of mice have been made, a lot of work has been done, so it takes a lot, and I'm thankful for all that fund. Just to acknowledge, Anne is the one who made these mice and published them in 2004. She's the one who discovered doubling MECP2 can be toxic. And then Amy Hezi and Yin Yao have contributed to the studies I shared with you in the past on the rescue. And then currently Samir is dedicated a big chunk of his effort to the biomarker studies, and Harini is also working in this area, and Wei Wang. And of course, I'm funded by Howard Hughes. And then all of our collaborators who help with computation, EEG studies. This is the regulatory element studies and our collaboration with Payman and Frank at Ionis. And this is a picture of the lab so that you get to see how we celebrated early on that we could go to the park and take our masks off. This is Samir, and this is Yin Yao, if you have not met them. And Ashley actually joining the MECP2 team to do the screens for druggable targets. So I hope you'll get to see these people in person. This is Wei, who does all our physiology studies. And also Manar has contributed to this area. And this is where we are. We hope to hold a meeting in next year or shortly after, whenever it's possible, so that we can give you hopefully in-person update.
There's a couple of questions, and I'm going to sneak in one question first. When you showed the graph of the MECP2 levels and RNA levels going down and then coming back up, and then it's 16 weeks, I think they were back up to kind of, back where you started. When you think about dosing, how often you would be dosing, because you wouldn't want to see this up and down in terms of the symptoms, right? You want to try to get the symptoms ameliorated and then hopefully stay there and not have this up and down. But because the symptoms lag,
right.
Do you think you would be dosing like maybe 4 times a year, 3-4 times a year?
I think that this is really an important question, and I should mention what we're using here are tool compounds, because this is animal work. The kind of ASO that will go into humans typically is clinical grade compound that the company will develop, and typically they've done them to where, as you mentioned, they will have a sustained effect for about 4 months, maybe 6 months. It all depends on the disorder. So the answer is yes, that will be a clinical grade compound that will have better durability. But I think we'll learn, that's why the mouse studies were important. We learned this dance. I want you to know that when the MECP2 levels went up at the RNA and the protein, the clinical improvement, it's still going on and it's still being sustained. So you don't want to actually dose earlier because you're going to drop it back again and you're going to confound this. What you want to do is time it exactly based on the mouse data, at least as you said, about every 4 months, because or 3 months, let's say, based on the mouse data, so that you're still getting the benefit. You dose again, it comes back down, but now there's no lag because you're still in that good place. And I think this is really why a biomarker is important, because I think you're going to really measure that and titrate and learn as you add dose upon dose upon dose. Does that buy you now, better sustainability of the effect? All of these things we're learning. And I have to tell you, no disease has been treated that is like MECP2. It's so sensitive in either direction, and really understanding all these things is really important. And the same is true for Rett. It's not like for Rett, if you overdose, you're going to see the effect right away. It's going to also take time. The benefit is also going to take time. So it's going to be really interesting to learn from these studies to fine tune our dosing. Great question.
Okay. So I'm going to start with the questions. There's one in the chat and then I'll move to the Q&A. So Carolina from Argentina has a question. Says, how do we ensure that MECP2 is responsible for generating epilepsy seen in our children, especially when duplication involves more genes, not just things?
Right. So if there's one thing I'm confident about, it's the epilepsy, because when you read Linda Vanesh's description of the set of cases, particularly the paper I believe published in the American Journal of Human Genetics, it's not only that they have the same kind of clinical seizure, but the EEG changes that we see in the animal models are ideal to the EEG changes we were seeing in patients. So I do believe that, and again, you'll see that there's only really 2 genes in the minimal region, IRAK1 and this one. We're not aware that IRAK1 can cause epilepsy if it's doubled. So we do believe, and then we see that we can correct this epilepsy in the animal. So I think that's one thing I would say it's more likely to be MECP2. And also I think what's really important, the reason I think most of the features we see are MECP2 is because many of these features really are the opposite of the loss of function. And you see them in the mouse and you see them in the human. Could there be other genes in the region contributing to phenotype? In some of the patients, I'm sure it's possible, but at least we can treat quite a few of the symptoms if we just target MECP2.
Okay, great. Let's see. So Burhan says, I know it's hard to estimate, but do you have a rough estimate about when clinical trials can start? I really am not at a privilege to make that claim or the date, just because this is really in the hand of our collaborators. And as I said, they're the ones doing a lot of the other background work, developing the ASOs for humans, testing them and so on. So I really cannot speak to that. I can only speak to what I'm doing, and we're doing the very last step that they need from us, get them as many biomarkers as possible. Are clinical studies done in Houston only? Do potential patients, will patients have to travel to Texas when first trial?
Only for the clinical readiness study. Those are done in Houston only, because those are very focused, very intense evaluation. As I told you, these will involve many clinical biomarkers that they are being looked at and involve both the duplication and the null. Our goal when it's time to do the actual clinical trial, or if we needed more patients, even for clinical readiness, is to expand. Definitely for the clinical trials, this will be a multi-site. Hopefully, strategically, these sites will be to make it easy on families to access them wherever they are. So no, the clinical trials will not be only in Houston. It's just this clinical readiness study required so many experts in so many fields that it was easier, and it was a small number of individuals. It was easier to do it here.
And I'll just add, because I get this question asked a lot from Rett families. Once whoever the sponsor of the trial is, whether it's an academic trial or an industry trial, once it has FDA approval to start, the protocol is published, is made public on clinicaltrials.gov, and we would probably have it on our website as well. And if your child meets the inclusion-exclusion criteria, then the list of participating clinics are included in the protocol, and parents can call the clinics directly to get more information or to schedule a visit to learn more about it, decide whether you want to participate. Okay, so Hannah asks, do you think one-time injection will be enough for the whole life? So no, this is not a one-time injection. It's not like gene therapy. This is ongoing.
Right. I think that just to continue this, Hannah, I think that eventually, technology keeps evolving. Right now, the way this is being delivered and so on, it's going to be multiple, we don't know whether it'll be twice a year, 3 times a year, we don't know that. But then eventually, hopefully, the therapy could be converted to be one time once you really know everything you want to know, and dosing and stability and all of that. There was a question in the chat, do I see major roadblocks on the horizon that will hold the project? Fortunately, I mean, my biggest scare was in the adult, if you gave the ASO, are we going to see acute depletion causing acute, severe, dangerous symptoms? That was my biggest worry about a roadblock, and that did not happen. To the contrary, using these ASOs and that acute depletion, the animals did fine. Yeah, of course they had the Rett-like features because we eliminated the protein completely, but at least they recovered from that. So I think we are now fine-tuning the steps, the last steps. So we're all committed to this.
Charlotte would like to know, and what cells are the biomarkers measured? Is it blood cells or CSF?
We're working on both. So we are working on both because eventually if we could do something peripheral, that would be much easier. But we have to validate using CSF to be sure what we look at periphery is reflecting what's in the brain. So we're working on both.
Bethany would like to know, when clinical trials start, will there be a placebo, or will all participants receive different dosages? I guess that's up to the FDA to some degree.
I think it's something we're going to have a discussion with the FDA. I think because there are a lot of clinical data on individuals with this disorder, we might be able to get by without a placebo arm, but I don't want to speak to that because we're not there yet, and I'm not the one designing the clinical trial.
Yeah, it's a bit premature, but also oftentimes if there is a placebo, there can be a crossover design so that people that receive the placebo first in a second phase can actually get the treatment. You don't know which, blinded as to whether you're getting placebo or drug first. Okay, David has a question. His 2-year-old was recently diagnosed, and he asks if mRNA protein rescue normalize at different rates, does this increase the challenges for ASO administration over time, since it is not a single dose?
No, it shouldn't. It's just that we needed to understand, the pharmacodynamics and the kinetics of the response. That's why we did the study. Just the fact the RNA changes first and then the protein changes does not in any way changes anything or the benefit from the therapy or the amount of dosing you're going to do. It just, we learned so much about this that we did not know before these studies. And that's important because you're going to the study with eyes wide open. You know exactly when to monitor. You know that you're going to have to run the trials longer. If nothing happens within a few, this is, 2 months in a mouse. I can't tell, the human may require a few more months. Whatever it is, at least we learned there is a lag time. And I think this is something that's going to be really important to know, but it doesn't in any way changes the benefit or complicates the study in any way.
Narinder would like to know if there's any way to anticipate how long it might take to find the biomarkers.
It is, I would have to say we're really dedicated to that. You're used to me, maybe Monica is used to me more than some of you, whereby, every year there's a new step completed and worked on. So our part, you can be assured the next 12 months, we are heavily investing and dedicated to that. Of course, then we turn things to our collaborators, but I think that we're doing everything we can to expedite this step. And you saw all the steps that happened just in the last, I want to tell you that the regulatory element story, the story about the reversibility in when we overdose, all of that happened in the last 24 months. And 18 months of those 24 months pretty much have been, tough. Yeah. Productivity-wise, but we managed to continue to work day and night to really get to there. So I think that the team is highly dedicated.
Yeah. And the other thing is, once you think you have a biomarker, it actually has to be tested, right, in a treatment to see whether it actually correlates and goes up and down with treatment. So that's another big step that has to happen.
Correct. Well, in addition to that, Monica, we do have the benefit of the male Rett individuals, where you don't have to actually do a treatment. You'll see if that same biomarker when MECP2 is too low, what it's doing. So we're studying those. That's why I'm thankful to the Rett families who participated in these studies.
And then someone, I can't pronounce the name, we'll check, I'm not sure pronunciation, but do you have a patient's age? Like what's the target patient age for doing studies? And I know that's a really tough question to ask.
If you're asking me now for the clinical readiness study, I think they were taking patients of all ages. If you're asking me about the trials, I imagine they will also take patients from all studies, patients from all ages into the study. I imagine, this would be the case.
Amy's asking how to get children involved in clinical trials. So that's, when the protocol is made public, the sites, clinical sites participating will be listed and you can call the sites directly. For the clinical study, they would contact the Houston.
Right, for clinical study, for the clinical readiness study, you can contact Davud Pelivan, who I think spoke last week or the week before. But for the clinical trial, when that's gonna happen, we need all of you, the more, the better. We would be so grateful to enroll as many as possible to really see if this is going to be beneficial, no matter what the size of the duplication, no matter what the type. We are mindful that different people have different levels. This is something Davud discovered, and he's now going to really characterize a large number of people with Samir. Samir is helping with that study to determine, because we may have to put people in different categories. Again, somebody with a blood glucose of 500, you give them a different insulin than somebody with a blood glucose of 300. So we're gonna figure all that out.
Hannah would like to know if you would expect the ASO treatment to help with intellectual disability.
If I look at the mouse data, the answer is yes. And if you ask me to predict what will happen in a human, I would say the answer is going to make these children able to learn. So I don't know that the children are going to immediately turn around and start talking and able to do things and think through things. I think you're going to consider them how a child who's 2 is learning things. And with some of these preparation and retraining, I really believe they will do better, but it's unknown. I'm just going with what data we have and with what knowledge I have about brain plasticity. I'm really very impressed how plastic the brain is.
So two people are asking about the drug screen that you're also doing.
Right.
Give a quick update on that, and we could also set up a webinar later.
Right. I think we need a little bit time on that. During the COVID, that was halted, and then we ended up focusing on some of these other things to move the ASO more farther. We also ran into a few challenges that sometimes the peripheral cells don't really mimicking what's happening in vivo, but it is now highly optimized. So maybe in a few months, we'll do a seminar that we have some target that if you manipulate them, you can reduce MECP2 levels. And we explored, this is what I showed you, the DNA elements. We're gonna look at those as well. So we're not, we're still focused on that. The effects they're gonna be smaller than the ASO effects, but it allows you, if we have something like that, to combine it maybe with an ASO, to always use a lower dose of ASO and combine a drug. I don't think a drug alone will ever get you to from 200% to 100%, but it may get you slightly lower to help with lowering the dose of the ASO.
Someone's asking if you could put Dr. Davud's email in the chat.
Sure. Just give me 1 second and I'll do that. I can continue to answer questions while I'm pulling it.
You are a good Tom is asking, as a parent with a recent diagnosis, is there anything we can do to help support the research, genetic reports, etc.? I guess one is if you can travel to Houston to do the clinical readiness study. that would be helpful.
Yes, and I think that also there are two things that would be helpful: the clinical readiness, but everyone on this call could also send blood samples to Dr. Pelivan for the study that looks at the rearrangement, the structure of the duplication, and the levels, because the more we know the levels of the protein in every individual, the better we know, are they going to be in high MECP2, medium MECP2, what dose of MECP2 they have. So that would be helpful, not only to the research, but that will be helpful to you. You can say, my child actually have 3 times the protein level, or 2 times the protein level, or 1.5 times. So I think this would be really helpful.
And the other thing, Tom, is you can help raise money for this research. A lot of what's been discussed today has been funded in part by families. So I'll put my email address in the chat, and yeah, and feel free to get in touch with me, and I can give you more information on how to help. Okay. And can you put, Dr. Zoghbi, can you put Dr. Pelivan's email?
I did. I put it in the chat.
Right. So, yeah, reach out to Dr. Pelivan, Tom, because it is a specific test, I imagine. So you can coordinate with him about that.
Yes. He'll just tell you what kind of tube the blood should be put in. And it doesn't have, it could be whenever you're taking your son for a blood test, they can draw the extra blood in a special tube, and he'll give you all the instructions.
Okay. Jen is asking whether there may be a cost to being in a clinical trial.
No, this will not be. We're subsidizing that actually here at the Institute, that particular arm of the study, to look at the levels of the protein.
Okay, I think we
would appreciate support for that. I mean, that's something I know Davud presented that. That would be great. That's the kind of research right now we don't have support for. Yeah.
All right, I think we've answered all the questions, and beautifully on time, I might add, 12:59. So Dr. Zoghbi, thank you so much for
you. And I really cannot thank all of you enough. I mean, from all the way from coming to Houston to meetings to allow me to see you and understand this disorder, to your patience as we do all the necessary steps for safety, your fund raising and contributions. I just think this is a partnership, and we're in it together. We are just as passionate to get to see the boys get better and the girls with Rett get better as everybody else. So we're really trying, and I appreciate all your support.
We'll upload the recording of this webinar to the Rett Syndrome Research Trust YouTube channel. If you go to our website, which is reverserett.org, click on the YouTube icon, and then click on playlists. There is an MECP2 Duplication Syndrome playlist with various recordings that we've done of webinars, and we'll add this one. Just give us a day or two to get it uploaded. Okay, thank you very much to everybody. Enjoy the rest of your week. Bye-bye.
Bye-bye.